morning. I hope everyone who's here knows I'm Matt State, Chair of the Department of Psychiatry and Behavioral Sciences. I'm delighted to welcome you to Grand Rounds today. For those of you who may be checking in via Zoom, there's still a couple seats down here in the front. And particularly our trainees, our residents, your entire class is here, so your personal absence is notable. In any case, people, I have to watch my sense of humor. That was a joke. But with some underlying meaning. So I am delighted this morning, truly thrilled, and I do hope that we have a large audience on Zoom this morning because we have the great privilege of having Dr. Anna Molofsky, an esteemed faculty member in our department, who is our Grand Round speaker this morning. So Dr. Molofsky did her MD PhD at Michigan, spectacular work there. We were successful in recruiting her to our research residency, and she thankfully is a proud graduate of our research residency, and did, again, spectacular work as a postdoctoral fellow with Dr. David Rowich, where she transitioned from her earlier work to focus on glia. She was successful in getting a K award and pretty much every other award that is offered to emerging psychiatric scientists. So in 2015, she became a faculty member, assistant professor, and since establishing her own group, she has really established herself as an international authority on the immune system and its impact on brain health and disease, which you're going to hear about today. So she's working on particularly microglia, the innate immune cells of the brain, and her group really early in her trajectory as an assistant professor has made several truly seminal, I think, groundbreaking discoveries around the role of microglial activity in synapse pruning via its interaction with IL-33, which again, I think you'll hear about a bit today. And then more recently, new mechanisms by which microglia mediate synaptic plasticity. And that latter work these were all highly collaborative, but that one I just want to point out in particular was with another spectacular scientist in our department, Mazen Kirbet. So her work is tremendously interdisciplinary. She has very close ties and really serves as a bridge between immunology and neuroscience, not only within our university, but really, as I say, nationally and internationally. She's achieved pretty much everything that you could achieve as an assistant professor, both in terms of extramural funding from NIH with multiple R01s. She's gotten multiple national awards. She's gotten international awards. This year, she received the 2024 Daniel Effern Basic Research Award from the American College of Neuropsychopharmacology. And then most recently, I believe maybe you got an additional award after, but we just announced that Dr. Molofsky was this year's winner of the prestigious Bose Biomedical Investigator Award. For those of you who don't know what that is, the one is given out per year across all of UCSF, all basic scientists, really looking for the most outstanding collaborative scientists who are most likely to make a fundamental contribution to advancing scientific knowledge and the largest kind of overall contribution to our scientific community. It is an enormous honor, and the department is, as you can tell, via me as a representative, enormously proud to have garnered a Bose Award. So all in all, Dr. Molofsky's been upholding the UCSF tradition, really started decades ago by Sam Barondes. She holds a professorship with his name, but he really established UCSF as housing the leading psychiatric physician scientists truly in the world in a clinical department, something that really was not the norm at the time. And I would add that in addition to a spectacular scientific career that's making a major contribution, not only to psychiatric science, but I believe you'll hear today, other neurological disorders, and thinking about the role of these mechanisms in health as well as disease. But I do want to add that in addition to her spectacular science, she is an extraordinarily active mentor who is a fierce advocate for advancing women and underrepresented communities in the basic sciences and in psychiatry more broadly. And on top of it all, she remains an active clinician where her focus is on dynamic psychotherapy with adjunctive medication management. In short, she is, I believe, and represents our department's highest aspirations about what it means to be an academic physician and psychiatrist. Anna, delighted that you have an opportunity to tell us about your work today. Thank you.
Thank you, Matt. As you can tell from that introduction, unflagging support from my department has been critical to me being here today. I think we all appreciate at these times that, you know, we may think that we're fancy scientists, but really we are working within a system. And it is so important that that system work well. Anyone who's done science in another country without the resources of the United States will appreciate that. And UCSF Psychiatry in particular has always been a place where people can ask these difficult questions about how do we mechanistically think about psychiatric disease. And Matt has continued that tradition and made it much stronger since he took over as chair. So thank you. So yes, you're gonna hear a little bit from me today about what should we think about when we think about inflammation in the brain. Is it good? Is it bad? Is it a way to think about new ways of treating psychiatric diseases? And I would argue that it's all three of those. The immune system is involved in so many different brain disorders, and I'm going to give you a few vignettes today of some of those. I wish I had more to say, concrete information to give you about psychiatric disease, and I'll allude to it throughout the talk. But I will also give you a sense of the progress that's being made in other neurological diseases that I think we as psychiatrists can learn from. Probably the touchstone neuroimmune disease is multiple sclerosis. And we're learning a lot more about how multiple sclerosis may be triggered by viral infections. And this is leading us to think more about post-viral neurological syndromes, right? The most famous of which is long COVID, which, as you probably know, we really have no idea what's going on, why patients with long COVID are presenting with brain fog, why it can exacerbate psychosis in some cases, or even exactly what the epidemiology of it is. I'm going to briefly mention, although not talk about schizophrenia, which is linked to the immune system because of genome-wide association studies that show that the class 1 MHC locus is a top hit in schizophrenia. And then I'll talk briefly about Alzheimer's disease, some of the work that my lab has been doing in animal models to think about what immunotherapies one could use to treat Alzheimer's disease. And finally, what is really my first love and what most of my lab has been working on for a long time, the concept of neurodevelopmental disorders, not focusing on a specific disease per se, but how can we understand how the brain develops and why the immune system matters so much for that process of development. So my background is on multiple sclerosis because I think it's a nice success story that really points to how beautiful it can be when you see a field beginning to come together. And briefly, multiple sclerosis is a disease where the myelin of the brain gets attacked by the immune system. And you see these patches, these lesions of demyelination, they come and they go, they're healed. And you know, once I believe you've had more than 3 of them, you're classified as having MS. One of the most exciting things to me that's emerged from the field of MS recently, and there's 2 of them, is the discovery that Epstein-Barr virus, which is a DNA virus, increases the risk for multiple sclerosis 30-fold. And I bring this up because I think we as psychiatrists need to think about when there are epidemiologic links that we see in psychiatric diseases, do we ignore them or do we say, hey, maybe there's something there, let's wait and see. In the case of Epstein-Barr virus and MS, people have been postulating for decades that people infected with this virus are more likely to develop multiple sclerosis. You know, we had all these soft signs, like people in the Northern Hemisphere are more likely to get MS, the kinds of things that we hear about a lot in psychiatric disease. We don't know whether to put much stock in it or not. And here, if you look up the literature in PubMed over the past, you know, however many years it was first detected in the literature, you see people saying, you know, multiple sclerosis and Epstein-Barr viral hypothesis. You see people saying Epstein-Barr virus and multiple sclerosis, no. So there was a lot of back and forth and disagreement. But there was no definitive link. And I would say pretty much everybody agrees now that EBV can increase the risk for multiple sclerosis until 2 years ago. And why? It's because everybody has EBV. This is the kissing disease, right? Mononucleosis. By the time you're a young adult, you've had it, right? And so this is kind of like in the 50s where epidemiologists said, if you're trying to prove a link between smoking and cancer, that's like trying to prove a link between sitting and cancer because everybody smokes and everybody sits, right? It's a similar thing here, but what these folks did is they had access to millions and millions of people in a military database that are getting sequential blood draws for HIV monitoring. And from those millions, they ended up with a few 100,000 people, young people that didn't have mononucleosis at the beginning of the study. And by following those people over the course of about 10 years, it became completely obvious that those that were seropositive for Epstein-Barr virus were much more likely to develop multiple sclerosis compared to those that converted that got infected later. So this, you know, and I won't go through all the calculations because I don't understand them, but this correlates to a 30-fold increase in risk. So this is unequivocal evidence, finally, that this virus does increase the risk of MS. Now, why does that matter, you might think? I mean, this is a bad time to be talking about this, but it brings up a question. Should we vaccinate against Epstein-Barr virus to protect against MS, right? This becomes a real possibility when you've identified a pathogen and you've proven an epidemiologic link. It's not a possibility if you fired 17 senior vaccinologists yesterday, but you know, we can hope that one day when things come back to rationality, that we can begin to have these discussions again. And there's another reason why this is really interesting, besides its therapeutic implications, and that is that therapies for MS have completely changed since I finished medical school to now. So much so that the literature is a little bit of a mess as to, like, you know, there's not like a clear set of practice guidelines. And a lot of that change happened from work happening here at UCSF. And one of those major discoveries is that blocking B cells, the kinds of immune cells that make antibodies, is a major new treatment option for MS. This was kind of revolutionary thinking at the time. And here's what's really interesting. Epstein-Barr virus is a virus that infects B cells. And so you can begin to see the field coming together where we really understand on a totally different level what causes this disease and how do we treat the disease. And this is where we wish, right, we could get for psychiatric diseases. And so you may know that the Breakthrough Prize was given to Steve Hauser, as well as the fellow who did that epidemiology study that I showed you a couple slides ago, just a few weeks ago, because B-cell immunotherapies and this understanding of this risk factor have so changed the landscape of multiple sclerosis treatment. And I just want to make one more point before I end this kind of introductory portion, which is that immune cells recognize pathogens via molecules that are on their cell surface called MHCs, major histocompatibility locus. And this kind of hints at a lot of what we learn about when we do genetics of these diseases. The MHC locus is so important because every human population evolved in a different setting and being exposed to different pathogens. So it's one of the most variable loci in the genome because it's tailored to the spot where each person evolved. And of course, the MHC locus is one of the major hits when you do genome-wide association studies of MS, as well as many other genes involved in the immune system. It's a big locus with 250 different genes, and so it comes up a lot. But the take-home point is that genetics, as well as all of the other data that I told you about, implicates immune genes in MS. And there's another disorder where the MHC locus, there's many actually, and there's one in which the MHC locus is particularly prominent, and that is schizophrenia. It's in fact the top peak of the Manhattan plot, even if you exclude a bunch of SNPs due to linkage and so forth. And this really means we need to think carefully about how the immune system is involved in schizophrenia. Now, this is not the same as MS, right? All of these other peaks are not immune genes, they are synaptic genes. But there is something about the way that we respond to pathogens and infections whose variability increases the risk of schizophrenia in some people. Most likely that's what this is telling us. I don't think that it's telling us there's a specific gene in this locus that causes schizophrenia, but it tells us that we need to understand what's going on in the immune system, particularly in early psychosis, of which people in the room could probably tell me a lot about. Epidemiology, although it's much less home run than what I showed you for MS, is out there, right? It's in the early stages compared to what we know about Epstein-Barr, but we know that a history of autoimmune diseases or a history of severe infection causing hospitalization increases the risk for a diagnosis of schizophrenia, and combined, there's a relative risk of 3.4-fold increase in risk. So pretty substantial. So what's really missing here is we don't know what the pathogen is, right? And until we know what the pathogen is, if there's only one, and being psychiatry, it probably won't be just one, it becomes very difficult to really prove these associations. And so the take-home points from this kind of introduction here is that neuroimmune disorders are complex gene bioenvironment interactions, that links between the immune system and brain diseases can take many, many years to definitively prove, but that once you know that, or even if you don't know that definitively, immunotherapies can be completely transformative. And before I finish this take-home point, I want to add an addendum to the take-home point, which is specifically about immunotherapies, which we have never used for psychiatric diseases. And kind of the long-term goal of my group, if I had to pick one, is to have one. You know, why can't we treat psychiatric disease with immunotherapy the way that we treat every other disease in the body? This is the fastest growing area of medicine right now. And one thing to think about is that immunotherapies can target disorders regardless of whether they are immune disorders or not. And one of the most famous examples, as always, is cancer. My slide is not advancing. Hold on. Oh, so melanoma. When I was a medical student, I was in a melanoma and renal cell carcinoma clinic. These are two deadly cancers that there were no treatments for. And the attending was like, you know, these cancers are kind of weird because every once in a while, a patient that has one of these cancers will spontaneously regress and the cancer will disappear. And we think it's the immune system. So that was the state of knowledge when I finished medical school. Today, last fall, a clinical trial of melanoma was finally completed, showing that an immunotherapy can, as said here, dramatically improve survival in this super deadly cancer. And I will say at the time, you know, when we were first studying melanoma, we had familial mutations and a gene called P16, which is the one we studied at the time. People made mouse models, they did this, they did that. None of that advanced treatment for melanoma, but immunotherapy has. So you can harness the immune system, which is already very biologically intelligent, to do what you want it to do in certain situations. In this case, to encourage killing of cancer cells. Any questions at this point? I'm happy to pause if that's better, or I can keep going. Yes, Damien. So the trial that just completed here was the final 10-year outcome. Oh, like how often do they have to have treatment? No, I don't know. I'm not an oncologist. I mean, I think it's multiple. Yes. I mean, the thing is, this kind of treatment, it's a nice example, but it's not the type of treatment we would probably use in psychiatric disease because cancer immunotherapies kill things. And so I've been collaborating with Wendell Lim, who's a bioengineer here. And when we first started talking, he would say like, well, I want to do immunotherapies for the brain. And I'm like, what, you want to kill things in the brain? Like, ha. But now I've come around, as you'll see. So it'll be different. And I suspect for the types of, I'll give you an example in the next section of what it might look like. And I think still the questions of, are you going to do it once? Are you going to do it a few times? Or is it going to be chronic? That's a really important one. So thank you for bringing that up. Other. So yes, so the next portion of the talk is about the immune system in Alzheimer's disease, which, as you can see here, is an example of immune cells surrounding an amyloid plaque in the brain. So Alzheimer's disease is damage to the memory centers of the brain. It's the most common form of neurodegeneration. The first hint as to the pathology of Alzheimer's came when Alois Alzheimer discovered that there are plaques and tangles in the brains of patients with Alzheimer's. And one of the reasons it's such a critical area of research right now is that 1 in 9 people over the age of 65 will develop Alzheimer's disease, and we're living much longer than we used to. And so it's becoming more and more common. My dad has it actually. And so there's a huge push to really target this disease. And we understand much more about the pathology now. So we know that there's plaques and there's tangles, but we now know that the plaques are caused by a protein called amyloid, and the tangles are caused by an intracellular protein in neurons called tau. And what we know is that amyloid, and this kind of gets to Damien's question, amyloid forms first. So amyloid plaques are building up in the brain 15 to 20 years before there are any symptoms at all. And at some point, the amyloid helps to trigger this pathological transition where tau, which you can see here in blue, starts to aggregate much more quickly. And that's what leads to this profound and sharp cognitive decline. So people have wanted to understand, well, you know, is amyloid really that bad? Is it not that bad? How come people have it, but they're not even that sick? And that's one of the controversies in the field that you might have heard about. So before I get to that, I want to introduce microglia, which Matt introduced. These are the immune cells of the brain, and they actually are 10% of the cells in the brain. So that's pretty striking, that 10% of the cells in the brain are actually not brain cells at all. They are part of the immune system. And here you can see some microglia, 1, 2, 3 of them, plus the processes of a few others. They're intermingled among the synapses in the brain. And they can actually take up synaptic proteins as the brain is turning over and remodeling. You can see some of those synaptic proteins inside of microglia. So we know now that microglia have very critical functions in remodeling the brain, keeping it healthy, cleaning away debris. And they also have a really important role in Alzheimer's disease. Because what they do is they cluster around those amyloid plaques. And here you can see 6 of them clustering around a plaque, and they sequester that toxic material, prevent it from leaking out and damaging the rest of the brain. And we know now, because the human genetics, again, back to GWAS, strongly implicate immune genes, that when you knock out these immune genes in the mouse, what happens is those microglia fail to do that protective job. So the key point is that the immune system is good. These microglia are protecting the healthy brain from plaques. So you can imagine that the better they do their job, the less likely that plaque is to trigger that tau accumulation that leads to decline. And I don't know if whoever here has ever seen me give a talk before, but I'm usually kind of a cynical person, right? But I'm here to tell you, like, I was at a meeting recently, I called my husband, I said, when we get Alzheimer's disease, there will be therapies. We are on the cusp of a treatment revolution. I'll tell you why. So first, you know, there was a lot of back and forth. If you read the New York Times, you would think it's all a mess. It's actually not. This study published in 2023 was the first to show that an antibody that targets amyloid plaques can help slow cognitive decline in Alzheimer's. This is huge. It's not a huge effect, but it's effective. So this means we can take that kind of discovery and make it better. And it also means that it's worthwhile targeting those plaques, and particularly before the decline of tau aggregation has begun. At the same time, and this is like the same year, biomarkers, blood biomarkers that can detect Alzheimer's disease before it gets worse are going to come into clinical practice. This is happening now. This detects levels of phosphorylated tau. This marker was like 3 times as good as a primary care doctor and twice as good as a neurologist that's detecting mild cognitive impairment that will progress to Alzheimer's. And people are also working on making antibodies better at getting into the brain. One of the main reasons they don't work as well as they could is that the blood-brain barrier prevents them from getting across, and there are techniques to do that. So you can see when a field is at the edge of multiple different advances that'll come together therapeutically. And so we're interested in understanding whether we can modify some of these existing therapies to harness the brain's own immune responses to optimize immune clearance of plaques. Can we make these antibodies not just stick to the plaques and get a little bit of clearance out, but actually help the microglia to compact those plaques? And this comes back to the immune signal, the cytokine that my lab really started on 10 years ago, called interleukin-33. And we've showed that this cytokine is really playing a critical role in the healthy brain. It actually helps to promote brain development. It helps to promote learning and memory. We've shown, for example, in the hippocampus that interleukin-33 encourages microglia to remodel the extracellular matrix around synapses. So it promotes synaptic plasticity. And that this is important for long-term memory formation, which we can study in these mouse models of contextual fear conditioning. And all this work was in collaboration with Mason Kierbeck, as you heard from Matt, and it's ongoing, though I won't talk about it today. Now, Dongkyu Kim, who's a very talented postdoc in my lab and also an expert in mouse models for studying Alzheimer's disease, began to look in a mouse model that develops human amyloid plaques. So it's expressing the human amyloid proteins, mutations, and it begins to develop the plaques, which you can see here in orange, pretty early in life, so when the mouse is 4 months old. And what we found is that if you get rid of this immune signal, interleukin-33, the plaques are much worse, both in males and in females. So IL-33 deficiency worsens amyloid plaques. So we need this immune signal in the healthy brain to protect the brain from plaque accumulation. How is it doing it? Well, as I hinted to you, the microglia have to compact plaques. What we've shown before is that this signal activates the function of microglia. And when you knock it out, the microglia are much less able to surround and sequester these plaques. Here you can see a single plaque, and you can see these microglial end feet, kind of like a dung beetle kind of wrapping it up. And so we thought, okay, well, if getting rid of IL-33 makes plaques worse, can we make them better by delivering IL-33? So that took a little bit of protein engineering, because this is a weird kind of a cytokine. So most cytokines are secreted. They have a secretory domain that gets them secreted out into the blood, into the tissues. This is why we can measure IL-6 in the blood and IL-1 and many things that people measure in depression, for example. But IL-33 is sequestered in the nucleus. And so to make it be secreted, here you can see it's localized in the nucleus of the cell. To make it be secreted, Dongkyu made a modified version that lacks this domain. And this version now you can see is not sequestered in the nucleus. It's all present throughout the processes. This is the green signal here. And this ends up being secreted. And so when we deliver this construct using an adeno-associated virus to express this in the brain of these mice that develop amyloid plaques, what you can see is that there's a marked reduction in plaque pathology in these mice. And even more exciting, we can actually test memory in these mice. This is called a Y maze. So the mouse likes to explore, right? It'll explore one arm and then it'll explore the other arm next. But when there's deficits in short-term working memory, the mouse will go over and over into the same arm. So that's called impaired alternation. And that's a metric of a deficit in short-term working memory. And you can see that mice that have these amyloid plaques have impaired short-term working memory. But knocking out, delivering this IL-33, this therapeutic cytokine, restores that working memory. And importantly, IL-33 is very strongly expressed in the human brain, also in the nucleus of these cells, which means we need to find ways to deliver it, to get it to come out of the nucleus if it's going to be therapeutic. But by the time you end up with end-stage Alzheimer's disease, it is profoundly decreased in quantity. So this raises the question of whether IL-33 augmentation could be a potential therapeutic strategy in Alzheimer's disease. So the take-home point here is that immune signaling can be protective, and it's critical for a healthy brain, and that delivering cytokines to the brain is a potentially powerful therapeutic strategy. And this is something that we want to apply to a lot of other cytokines that we've identified that I won't have time to talk about today. But any questions at this point or thoughts? Yeah, I'll continue. So the last point, the last part of the talk is about the immune system and its role in brain development. And so you'll notice here that I won't be talking about mouse models. I'm mostly talking about how the brain develops in a healthy setting. And just to remind you, the brain is developing up until we're about 25. And The process of brain development begins with the birth of neurons and glial cells, the migration into the cortex. They start to elaborate their processes. And for most of postnatal life, childhood, adolescence, it's the formation of synapses. So here's a single neuron that has over 8,000 excitatory synapses. Each of these green dots is a synapse. And here's that same neuron, which has over 1,000 inhibitory synapses. And the balance of excitatory to inhibitory synapses is what determines so much about brain function. We know that there's many models that autism involves an imbalance of excitation to inhibition. This is probably broadly applicable across diseases. And glial cells, which is what my lab often studies, like microglia and astrocytes, play an important role in that process of synapse formation. Now, what about the immune response? So there's one more teaching point here, was that there is no one immune response, but immune responses come in different flavors. And I think we all intuitively appreciate this, right? When we have a viral infection and we have an allergic response, those are two completely different types of responses, right? One makes us feel sick and shiver and cold. The other makes us sneeze and have, complications of allergies like asthma, airway hyperreactivity. And there's more and more data, which I won't talk about, showing that these have different impacts on the brain and on behavior as well. And these are different types of immune responses that are driven by different upstream signaling pathways. Viral infections are driven by interference, right? Interferon signaling is what makes you feel sick. It's also what fights off a virus. It's also a key feature of why some people get sicker from COVID than others, for example. These are very potent cytokines, whereas allergies are driven by a whole different set of cytokines called interleukin 4 and 13. And so in the healthy brain, in the healthy immune system, there's a balance of this going on all the time. There's a little bit of these cytokines, and there's a lot more that get engaged when we get like a real serious infection. So it's not that there's no immune response, there's no inflammation in the healthy brain. It's just that these are balanced and tuned to the appropriate situation. And our lab has shown that type 1 immune response, this antiviral immune response in the healthy brain, helps to eliminate damaged neurons. I'll show you a little more about that. And that conversely, this type 2 or so-called allergic immune response helps to promote the formation of inhibitory synapses. And I won't be talking about this today, but I can imagine that delivering a cytokine that promotes the formation of inhibitory synapses could be very therapeutic in the setting of childhood epilepsies. This is something that we're thinking a lot about now. And so there's an example of how an immunotherapy could help to treat a brain disorder, even though I do not believe that epilepsy is caused by a deficiency of interleukin 13. But in the setting of this so-called antiviral immune response, this is something we're doing a lot more with because there's so much evidence from so many different domains that these types of immune responses are pretty important for the brain. And what we have found, and this is already published, is that microglia, which come in many different types, have a particular subtype that seems like it's responding to viruses all the time, even though there's no virus present. This is a physiologic response. And when we look at these microglia, I showed you a picture of some before. These are very lacy cells that are contacting synapses. This unique subtype of microglia is eating entire neurons. You can see this one here. It's got two arms, right? One of them is in the process of eating a neuron, and the other one is in the process of chewing up a neuron. It's eaten two neurons at once. So we call these super eater microglia. And we're trying to figure out, why would you want to eat neurons in the healthy brain? You can see evidence of this in a zebrafish model. So some of my lab has been working with fish, which are transparent, so that we can directly image into the living brain. And here you see a healthy microglia that's just touching the neurons in red here, and they kind of have this surveillance motility. But when we drive these antiviral immune responses, microglia can actually eat. And you can see here, take away an entire neuron, chomp, chomp, chomp. What we ended up finding in this study is that microglia are necessary to eat some neurons that have become damaged and are expressing stress markers like DNA damage and so forth. And so, it's not just remodeling synapses that's happening when the brain is developing. It's getting rid of some neurons that may not be as necessary as others. And that this process is critical for excitatory inhibitory balance. So when we get rid of these pathways in microglia, we end up with too many excitatory neurons. And we see this very robust tactile hypersensitivity in the mice, which when we tickle their whiskers, we see aversive stimuli responses to these tactile hypersensitivity. Now, humans don't have whiskers, but that's okay. Tactile responses are also a really core feature of many neurodevelopmental diseases. And it's worth thinking about how an immune pathway could help to regulate sensory responses. So, okay, so these antiviral immune responses are healthy at low doses, but can you have too much of a good thing? And this is where we start to think about animal models of disease that could teach us something about these immune responses. And I'm not going to talk too much about this, but one of these is Down syndrome, which actually it turns out is a mild interferonopathy. Because many of the genes that drive this immune pathway are on chromosome 21. So there are very rare diseases called type 1 interferonopathies, and these include a Cardi-Gutierrez syndrome that lead to very severe intellectual disability, autoimmunity, brain calcifications. But Down syndrome is more of a mild type 1 interferonopathy, where you get some of these same features, but some features which are different. And Sarah Anderson, who's a postdoc in my lab, has been studying a mouse model where they took a whole chunk of a mouse chromosome that has most of the same genes that are present on chromosome 21 of the human and added it into the mouse so that this mouse basically has essentially what is a version of trisomy 21. And she's observing that the brain of these mice is very different with respect to their immune responses. When she does single cell sequencing of the brain cells, she sees that interferon responses are ramped up in these brains, and particularly in microglia. So all of the classic genes that we see upregulated when a microglia is about to go eat a neuron are also upregulated in the brains of these mouse models of Down syndrome. And Sarah's trying to understand to what extent this drives some of the cognitive impairment in this neurodevelopmental disease. But I also want to talk briefly about another setting where you may have too much of a good thing, which is early life viral infection, right? So if we have antiviral microglia getting rid of neurons, what happens when you have a ton of antiviral microglia when there's been a viral infection? And one of the reasons this matters, and coming back to the epidemiology that I mentioned at the beginning of the talk, I would say this is not home run epidemiology, but it's hints, right? There's a lot of different hints from many different kinds of studies suggesting that early life viral infections can increase the risk for neuropsychiatric diagnoses across a broad range of diagnoses later in life. And the idea is that there's a critical window of brain development, and this could be in utero or early postnatal life, where the brain is particularly sensitive to these viral insults. And reminder, the immune system is also underdeveloped at this time, right? So we don't have the same robust immune system that can defend against these pathogens later in life. And so Richard Jin, and I may go lightly over this because some of you may have already seen Richard's talk, but he is an outstanding postdoc and psychiatry resident about to start his child fellowship, who has been studying a mouse model of early life viral infection to understand how viral infections impact the brain. And the question is, if they're eliminating neurons like we would speculate they are, is this causing a kind of a hit and run damage to the developing brain? And so he developed a mouse model of a very common childhood pathogen. This is the one that causes hand, foot, and mouth disease. This is not like a brain infection. It can cause meningitis, but it's also an infection that most children will have, kind of like mononucleosis, Epstein-Barr virus. And so he infects young mice intragastrically, so nowhere near the brain. The mice actually do get virus in the brain. Does this happen in humans? We don't know. Nobody's probably ever checked. It may not. And the mice get sick. They have illness symptoms that resolve by the time they're about 14 days old. And then they look fine. So all of the mice recover. All of the mice are healthy. I would say this is probably a kind of a viral infection that would lead a baby to be hospitalized for a couple of days and then released home. And what we can see here, and this is using a technique called spatial transcriptomics, which can look at the expression of many genes at once in their spatial context in the brain. So here you can see the cortex, the hippocampus, the thalamus. What we see is that the interferon response genes are going crazy at about right when the mouse is pretty sick. But when it gets better again, it goes almost completely back to normal. So this is a transient response. When we look at these areas of infection, we notice that they're clustering around areas of the cortex. So it's not every part of the brain that's susceptible to this virus. And when we look inside those little hotspots, we can see microglia eating things. It's hard to tell what they ate because they digest them up so fast, but they could be eating neurons. They could be eating other immune cells. In fact, we think that they are to some extent eating other immune cells because when you have a viral infection and you get virus in the brain, immune cells that are not supposed to be there come in. And the most famous of these are T cells. And this is a marker here in green that can show T cells are actually infiltrating into the brain of these mice. And I think that microglia may be eating some of them. We have kind of a balance. We have kind of the Godzilla versus Kong situation of different immune cells facing off against each other to both help control the virus, but then get rid of them again so that we don't have persistent inflammation in the brain. What's striking, though, is that these mice get better. The immune response seems to almost completely go away. And yet, there are persistent behavioral deficits in the mice. And what we keep seeing over and over again is these sensory deficits. This is a beautiful assay that was developed by Catherine Dulac, who was our speaker earlier this year for the, what's that lecture called? The Barondes lecture. She developed this cool assay to be able to observe the mice without having to train them or do anything fancy. You just look at them, and they have a choice to go through a fuzzy tube or a tube that's not fuzzy, and you watch them. And so when Richard did this, he could see that mice that had been previously infected and got better again had a preference for the fuzzy tube. So there's a change in what they prefer, their tactile sensitivity, that is different, even though they look totally fine and recovered from the infection. And the converse experiment can be done where you tickle their whiskers with a Q-tip, and the mice that had been previously infected have a marked tactile hypersensitivity. So there are persistent changes in brain and behavior that last long after the viral infection is improved. And what Richard is trying to figure out now is why. One possibility is that the neurons themselves are fundamentally changed in some way, and we have hints that this might be the case. So here you're looking at a spatial transcriptomics experiment that Richard did, where he's analyzing what genes are still different after the mice have completely recovered. And what was really striking was that immediate early genes, the genes that indicate that neurons are hyperexcitable, remain really high all the way into adulthood. And you can see here by levels of FOS expression all throughout the cortex that there's diffuse hyperexcitability that persists into the brain long after a viral infection has resolved. There's also a change in the immune system that, although it's subtle, is detectable. There are T cells that get stuck in the brain and then can't get out again. And this may not look like a lot, but for a cell that's not supposed to be there at all, it's pretty striking that there are T cells still persisting in the brain all the way into adulthood. And so what we're trying to figure out here is exactly what's causing what. Why is the brain hyperexcitable? Are those T cells making it hyperexcitable, or are they actually protecting the brain from hyperexcitability? So more to come on that. But what I want to point out is that this study makes it clear that viral exposures can also trigger hyperexcitability in disordered neurodevelopment. Many people have shown that mouse models for autism can lead to a broad kind of diffuse hyperexcitability, and we need to understand much more about what we mean when we say hyperexcitability, and that's where, you know, there's folks in our department with a lot of expertise in actually understanding what that means and looking at neurons firing in real time. So all of that work still needs to be done. But you know, those mouse models of these monogenic disorders that strongly increase the risk for autism suggest that genetic influences can have an impact on hyperexcitability of the brain. What this study shows is that the immune environment and a history of infection can also have an impact on hyperexcitability and lead to disordered neurodevelopment. And so when we think about complex disorders that can have many causes, we need to consider each of those one by one to understand how these many different triggers for disordered neurodevelopment could converge on similar phenotypes down the line, with the goal of one day understanding different subtypes of disease, understanding that one disease can have many different causes. And I think this is a broad goal for many of us in this room. I'm not saying anything new. So that's the end of the talk, per se. I just want to point out that the take-home point is that the immune system shapes our brain. If you've seen this joke about types of scientific papers, there's like, we scan some undergraduates, we put a camera somewhere new. The one that resonated with me is the immune system is at it again. If you think that that's not applicable to the brain, I hope you now appreciate that it is. The immune system is applicable to every tissue in the body. It is a core regulator an integrator of biological functions. And I think that we ignore it at our peril. Particularly in the setting of psychiatric diseases, we are still so desperate for causes and for new therapies. So thank you for your attention. Happy to take questions. Thanks so much to my lab, which is in the beautiful Weill Institute for Neurosciences. Always has beautiful sunsets. To my collaborators, including Ari Molofsky, he's actually my husband, but he's the immunologist that helped us to bring together this kind of neuroscience and immunology focus, and to my funding sources.
Well, wonderful.